Hello and welcome. Today we are going to demonstrate how to download public raw NGS data from the NCBI SRA database using the direct manual download method. Raw sequencing data can be downloaded from the NCBI SRA database in multiple ways, including direct manual download from the run browser of the SRA database, using the preface command followed by the FASTQ dump command from the SRA toolkit, and using only the FASTQ dump command from the SRA toolkit directly without using preface first. Since this course focuses on 16S rRNA Amplicon sequencing data analysis, we will demonstrate how to download raw 16S rRNA Amplicon sequencing data. Now let's look at the first method for downloading raw sequencing data from the NCBI SRA database, that is direct manual download from the SRA run browser. In this approach, we manually download sequencing data directly from the SRA web interface using a web browser. This method is simple and beginner friendly and is useful for downloading small sequencing datasets directly through the web browser. However, this method has some limitations. The SRA run browser allows limited downloading of sequencing data over HTTP. Only one sequencing run can generally be downloaded at a time, and the dataset size is typically limited to less than 5 gigabases when downloading through the browser interface. Now let's move on to the live demonstration. First, we need to open the SRA run browser. If you already know the address of the SRA run browser, you can directly copy and paste it into the browser. Alternatively, you can go to the NCBI SRA database. Under Tools and Software, you will find the SRA run browser link. Click on this link. This is the SRA run browser interface. Here we can directly search for a sequencing run using an SRA run accession ID. Now let's enter an SRA run accession ID in this search box. For example, SRR 3754459. We can use this SRA run accession ID. After entering the accession ID, click on this search button. Now the run browser displays information related to this sequencing run. It contains sequencing information such as read length, SRA experiment details, sequencing design information, as well as biosample and bioproject information. For example, here we can see the read length, here we can see the experiment detail. Here the design is 16S rRNA gene V3 to V4 amplicon sequencing from human stool samples. As we scroll down, we can see the biosample and bioproject information, as we can see here, associated with this sequencing run. These metadata provide additional biological and experimental context related to the sequencing dataset. Here we can see FASTA or FASTQ download option. Click on this FASTA or FASTQ download tab. To download, click on this FASTQ under the download option. The download is completed. Here we can see the downloaded file. One important thing to remember is that if the sequencing run is paired-end, generally a single interleaved FASTQ file will be downloaded from the run browser. An interleaved FASTQ file contains both read1 and read2 sequences together within the same FASTQ file. The forward and reverse reads appear alternately in the file rather than as separate read1 and read2 FASTQ files. This means that paired reads are stored sequentially within one FASTQ file instead of being separated into two independent FASTQ files. We can decompress or extract the file. Here we can see the extracted file. Another important thing to remember is that the FASTQ file downloaded from the SRA database may contain sequence headers that differ from the original headers generated by the sequencing platform or from those originally submitted by the researchers. This happens because SRA database often regenerates or reformats FASTQ headers during sequencing data processing, conversion, and export. However, the actual sequencing reads remain unchanged. As we can see here, the SRA run ID is added in the header. Now let's also look at the structure of the downloaded FASTQ file. As this is a paired end sequencing dataset, the downloaded FASTQ file is in an interleaved FASTQ format. This means that read1 and read2 sequence are stored together in a single FASTQ file. As we mentioned previously, they appear alternately, on read from the forward direction followed by on read from the reverse direction. As we can see here, this is read1 and this is read2. Here read1 and read2 records are arranged one after another in the same file. This is different from standard output formats where read1 and read2 are stored in separate FASTQ files. There are also other ways to access the run browser. In most cases, the run browser is accessed through the Entrance SRA experiment page. As we demonstrated in the previous lecture, we can access the SRA experiment page in different ways, such as direct searching using an SRA experiment ID or via a bioproject ID. First, we will have to go to the NCBI SRA database. If we already know the bioproject ID, we can search it in the SRA database. For example, PRJNA 1400113. Now click on search. Here we can see all the experiments associated with this bioproject. As we can see here, if we click on any of this experiment, we can see the details of that experiment along with the associated metadata. As we scroll down the page, we can see the associated metadata. At the bottom of this page, we can find the SRA run ID and its associated metadata, as we can see here. So instead of directly opening the run browser page, we can first open the SRA experiment page from the SRA database. Then click on the associated SRA run accession link to directly open the corresponding SRA run browser page. For example, click on this accession ID. Here we can see the run browser page. From here you can again use the FASTA or FASTQ download option to retrieve sequencing data as shown earlier. Here we can see the download option for FASTQ. Although this method is simple and useful for beginner, it is not ideal for downloading large sequencing projects or multiple sequencing runs. For large datasets, command line tools such as the SRA toolkit are generally faster, more stable and more efficient. So to summarize, in this lecture we demonstrated how to download sequencing data manually using the SRA run browser. We learned how to search sequencing runs using SRA run accession IDs as well as other accession IDs, and how to download sequencing data directly through the browser interface. That's all for now. Thank you everyone.